Hey, welcome back to Biotech Blueprint. I'm Katarina Rosnick, and today we are talking to a rare second-time founder who built and sold one biotech to Big Pharma and is now taking another into phase 3. Dr. Josep Bassaganya-Riera is Executive Chairman, President, and CEO of NImmune Biopharma. NImmune's lead program, omilancor, is once-daily oral gut-restricted therapy for ulcerative colitis, with follow-on potential in Crohn's disease and other inflammatory diseases. NImmune also leans heavily on a computational platform that we'll discuss called Titan X for discovery and development decisions. So we're going to focus today on what actually works in 2025, how to build biotechs in a tougher market, whether oral immunology can clear today's higher efficacy bar, and where AI belongs in the budget line instead of a buzzword bin. Joseph, welcome to the show.
Thanks for having me, Katerina.
My pleasure. It's great to have you. Thank you. Yeah, so I'll get straight into it. As I mentioned, you sold a company to a big pharma, to AbbVie, in May 2024. And so when you look back at this acquisition and sort of the entire Landos arc, what did you learn about big pharma actually valuing, and what did that teach you as a founder?
Yeah, first, a point of clarification. I think you've characterized me as a second-time founder. It depends on how you count that.
Right.
Since before establishing Landos, I created biotherapeutics. I guess that would count at the very least as a third-time founder.
So serial. I should have said serial founder.
Well, no, serial sounds maybe too rough, but at the very least third. But so when I look at Landos and the acquisition by AbbVie, I think what it shows is prioritizing a strong founder teams with a shared scientific vision, with an interest in addressing those unmet clinical needs of patients. Our focus, our priority has been on patients with inflammatory and autoimmune diseases, starting with ulcerative colitis and Crohn's disease. But clearly, the small molecule therapeutics that we've developed have the potential to scale up to other indications such as lupus, rheumatoid arthritis, psoriasis, atopic dermatitis, and so on and so forth. And so my team was very supportive of my vision, and we were grounded on science-driven, patient-oriented, from early discovery to clinical development to this exit. And I think that this is a key ingredient. This team basically was involved in inventing the product pipeline that eventually AbbVie acquired. And let me say, AbbVie acquired the early stage assets. They had the assets that were less de-risked, and we were able to keep the assets that were more advanced, including omilancor, NIM1324, and others. And so that same team that had from inventors to regulatory people to developers to financing people that were successful in helping us IPO in 2021 are now part of NImmune Biopharma, which is the company that I'm leading today. And I'm very glad that those people have remained loyal to the original vision that first established Landos, and now it's continuing under NImmune.
That's interesting. So most people stayed. And what did you do differently, I guess, this time, if anything, to build and run the company, that is now NImmune compared to Landos?
So I think it's not about doing things differently when switching from one company to the other. I think every single day within Landos, within biotherapeutics, within now NImmune Biopharma, we are adapting to the changing conditions. Like the current climate is very different from 2021, and that was very different from 2019. And so you need to adapt, evolve. What we are doing under NImmune is adapting those core principles of science-driven, patient-centric approach, which were so prevalent in Landos, and we are combining them with advanced advanced computational methods. We've integrated what we call the Titan X platform, which is an AI-powered system to accelerate drug development, make predictions, and help from target discovery to preclinical testing to clinical testing in a way that eliminates, to some extent, the trial error component that's so prevalent in this line of business. So my team is aligned with this approach across discovery, translational, clinical development, and I believe that alignment is absolutely essential. Now, I think one of the aspects on how you change as you adapt, have better technologies, have novel mechanisms of action, maybe the next generation of drugs are in development, you also need to recognize that sometimes changes are needed in the team. And while I would say that 90% of the team is the same, switching from one company to another also provides an opportunity to basically refresh the team, have infused new blood, and also eliminate maybe some people that have become dead weight and no longer contribute to the mission. So I think as a CEO, and I know that's probably not the most popular thing to say, but that's the reality of our business, and that's ultimately what makes these businesses successful, competitive, and achieving the mission, which is helping these millions of people with devastating and widespread diseases.
Yeah, absolutely. Thanks for being honest. What do you think was like the biggest, I guess, or what is the biggest challenge that you face now in 2025 that you maybe didn't face in like 2019? If you can do just a little comparison, what it's like now compared to just a few years ago?
Yeah. So 2019 was the peak of the biotech market, right? So the capital markets were absolutely open. Now move forward 2025. We came into the year with I would say a healthy dose of optimism as to what was going to happen. Then by the middle of the year, it seems like the biotech capital markets unfortunately collapsed, and then we are now bouncing back. So it's been like kind of a roller coaster, whereas 2019 was so much more stable and open, and so you just need to adapt to those conditions. But that also offers an opportunity to maybe identify better partners. There may be some partners, some investors that are more interested in the shorter view on things, and maybe being a more patient allows you to interact with those that have a long view on things, which allows companies to accrue more value. So I think that those are some of the considerations, but the fundamentals haven't changed, right? How you run the company, how you build the clinical pipeline, how you establish the right entrepreneurial culture in order to be successful. We are a small biotech, and we are competing with big pharma. Some of those big pharma maybe down the road will become acquirers, but they have limited resources, and we need to be much more creative, faster, in order to be able to accomplish things.
Okay, thanks. So let's layer in the science. So the LNCL2 protein, which you're focusing on, is expressed on multiple immune cells, including regulatory T cells, which are important in IBD. And they're often dysfunctional or numerically insufficient. And so at high level, what is LNCL2 doing in IBD, and why is the gut-restricted oral approach that you're taking attractive today?
Yes, LNCL2 is a novel target that we've identified, we've characterized, we even have identified the natural ligand. You could look at LNCL2 as an internal thermostat that responds to inflammatory and metabolic distress. And then when the right molecule, either the natural ligand, which is abscisic acid, or our drugs bind and activate LNCL2, it triggers a series of downstream events that change metabolism and change immunity. On the metabolic side, what LNCL2 does is favor mitochondrial metabolism. as opposed to glycolytic pathways. As you may know, glycolytic pathways are more pro-inflammatory. Mitochondrial metabolism is more anti-inflammatory. On the immunological side, it makes immune cells more anti-inflammatory. You mentioned regulatory T cells. Regulatory T cells are one of the cell types that express LANCL2 in highest amounts, but also macrophages express it within the gut, epithelial cells express it, and on the immunological side, the expression of LANCL2 has been associated with the ability of regulatory T cells to exert their anti-inflammatory activity. When LANCL2 is impaired or not active, LANCL2 cells cannot induce immune tolerance, cannot suppress excessive or dysregulated immune responses. When LANCL2 is active, either through the natural ligand or through our drugs, then that pathway triggers a cascade of events that provides anti-inflammatory responses, provides healing, and disease protection. So LANCL2 is a central theme mechanistically in what NImmune is currently doing. And we are very excited because there's a paucity of mechanisms of action in inflammatory and autoimmune diseases, and particularly in ulcerative colitis and Crohn's disease. It seems like every drug that is in the market is a variety of JAK inhibitor or cytokine modulating drug. There's also a paucity of drugs that target specifically the gastrointestinal tract. And so omilancor checks a lot of boxes that we believe are going to be important in helping address those unmet needs of patients.
And so the oral route is better for this type of drug, basically.
That's correct. So omilancor is an oral, once daily, a small molecule therapeutic. And small molecules have been around for a long time, but it's amazing how powerful they are. There's billions of dollars connected to small molecules. You may have read, Galapagos was running a gene and cell therapy program until recently. They shut it down, and they redirected their efforts back to small molecules. So they went back to the essentials, the fundamentals, what is known to work. So, and also it's from an affordability perspective. You can think of omilancor as an opportunity to provide that extra benefit because it's a 4-step synthesis. So it's from a costing of production perspective, very different than producing a biologic. And so it provides an opportunity for a low cost of goods, which eventually we hope that some of that will trickle down to patients.
Yeah, and also it kind of makes me think it's sort of fixing the root immune defect instead of broadly suppressing immunity, which some of these other drugs do, which definitely is very clinically meaningful. So I guess on the translational side, how do these mechanistic insights guide your thinking in like endpoints and patient selection in the clinic?
Yeah, let me first address a very important comment that you have made, the complexity of the immune system. So the immune system is a massively and dynamically complex system. I studied the immune system for 20 years before I started to create biotech companies, and I did that in academia. I used advanced computational methods to do that. But if an important thing that we learn about the immune system is the redundancies that are built in how it operates. So most of the drugs in the space are trying to neutralize a single cytokine or a single pathway, right? And what we know is that the immune system has the capability to adapt that. And so, for instance, if we shut down TNF alpha, there will be other pathways that will be compensatorily upregulated to still provide that level of high inflammation. So that's a huge difference mechanistically. Here we have a new mechanism of action, which is a multi-pronged approach that targets immunology, that targets metabolism. And as you put it, it gets to the root of the problem. If part of the issue of IBD patients, if part of the issue of lupus patients is that they have insufficient number of regulatory T cells, if they have regulatory T cells that are dysfunctional or impaired, then being able to kickstart those regulatory T cells and make them functional again is more powerful and has the potential to provide greater therapeutic efficacy than just neutralizing an inflammatory pathway, which is most of the drugs in the IBD space is what they are doing. And I believe that the reason why there's so much switch in the treatment paradigm, going from one drug to another, is because, of course, those approaches work for a period of time, but then they cease to work, and then the gastroenterologist needs to prescribe the next medication. And it seems like every layer of prescription is not necessarily more effective, but what definitely happens is that it's less safe, right? So the most advanced drugs have black box warnings. Some of the drugs in the space have cancer, infection, and death, as opposed to, GI targeting drug that has, from a safety and tolerability perspective, a placebo-like profile. So yes, getting to the root of the mechanism, making sure that mechanism doesn't have toxicities connected to it is absolutely critical.
Yeah, so let's connect this scientific piece to the clinical program. As we mentioned, your drug omilancor is in phase 3 clinical trials right now. So sort of looking back at, the earlier stages, phase 1, phase 2 data, what are some of the lessons that, you carried forward into your phase 3 program?
Yeah, it's very important. First and foremost, as I just mentioned, we wanted to move forward a drug that is safe. And the safety profile of omilancor is absolutely unrivaled. There's no rivals in the space. It's placebo-like, I believe, in phase 1, because the GLP tox in animal studies had shown no dose-limiting toxicities, no issues whatsoever. The FDA allowed us to go to almost 7 grams in the highest MAD cohort, which is way beyond what was needed from an efficacy perspective, which was about 400 to 500 milligrams, and we didn't see anything concerning, anything that was different than placebo, and that was then validated in our phase 2 clinical trials, both in ulcerative colitis and Crohn's disease. So I think that the phase 2 has provided the clinical insights on safety, tolerability, and more importantly, dose selection and efficacy. Obviously, we want to move forward drugs that are efficacious. If you look at the space, there's drugs that are very popular and are very profitable, I would say, for the companies that are commercializing them. But if you look at what they accomplish, for the most part, on average, is that they work in 1 every 5 patients. Yeah? That's what most multi-billion-dollar drugs in the ulcerative colitis and Crohn's disease space are accomplishing. Our drug, omilancor, we are confident that can work at least 1 in 3 patients. So that's an improvement. And then there's an additional layer. We've been able to identify a gene signature which is predictive of response to treatment. So basically a biomarker that could be utilized to identify those patients that are the best candidates for receiving omilancor. And when we do that, we can get down to half of the patients. So 1 out of 2 being successfully treated with omilancor. And so if you compare it to 1 out of 5, 1 out of 3 is quite good. 1 out of 2 is much better. So start with the safety, advancing to efficacy in the general patient population, and then being more targeting, basically utilizing the buzzword that is utilized these days is precision medicine, right? So how do you provide those therapies that are tailored to the needs of patients? And so in our case, obviously, because omilancor is so safe, it can enter the treatment paradigm from mild to moderate to severe. And so how do we narrow it down? We have the possibility to narrow it down based on that gene expression profile that we believe can be used as a biomarker of response to treatment. And we've seen in our clinical studies that we can predict those patients in about 83% accuracy. And once we've identified that, we are very confident that omilancor will do exceedingly well in that patient population.
Yeah, that is fantastic. And you kind of answered one of my follow-up questions, which was sort of what matters most to the payers and like to the FDA. And I think that especially to patients is it's this kind of the future maybe of big pharma and of like treating diseases is identifying people in which the drug is going to work, right? That they don't have to go on the drug and see if it works or doesn't. It's just you have a biomarker that you can kind of identify ahead, which is very valuable in my opinion. Yeah, so I guess we can now move on to, you mentioned Titan X a few times. We could kind of talk a little bit about the AI side that you are developing at your company. Before we go a little deeper, can you just give us like a quick version on what is Titan X and what decisions in R&D it does to helps you make?
Yeah, absolutely. And if you allow me, before I even talk about Titan X, I'll provide some background on how Titan X is even possible.
Yes, please. Thanks.
So, so in my previous life, when I was a professor and I was studying the immune system at the more fundamental level, I was successful bringing together a team of software engineers, computer scientists, clinical trial people, immunologists, under a program called Modeling Immunity for Biodefense. There were 4 nationwide centers that were announced, I think it was 2009. There was a competition, a nationwide competition, where NIID, one of the institutes of NIH, said, no, we have allocated this money. We believe that we need to study the immune system in different ways. The traditional methods of studying the immune system are slow, and that was actually that appropriation was orchestrated in the context of the aftermath of 9/11. You may recall that 9/11, there were some envelopes with anthrax that were circulated with some of our congressmen. That was a stimulus for those people to allocate funds to basically be able to have faster responses to potential bioterrorism threats. And one of the ways of doing that was utilizing computational models of the immune system, AI, high performance computing, in order to be able to understand what's going on and drive this. So I put together a team, and we were successful securing one of these centers. It was $12 million program. There were 3 other centers. So mine was actually the only one that was not located in a medical school. There was one at Mount Sinai School of Medicine. There was one at University of Rochester, again, the School of Medicine, and another one at Duke University, the School of Medicine as well. And our focus was the gastrointestinal system in the gut. And so we built the first large-scale computational and mathematical models of the gut immune system geared towards infectious disease. Again, that was not a program related to inflammatory and autoimmune diseases, but in the context of that program, we started to build the tools, the methods, the skill set in order to be able to develop those advanced computational systems. That project started in 2010. And I think that was a paradigm shifting project. It led to multiple products. It led to a tool called ANISI, Enteric Immunity Simulator. It was an agent-based modeling approach. We also built some tools that ordinary differential equations. And again, that is not Titan X. But it turns out that some of the PhD students that I had trained during the time that I was leading modeling immunity for biodefense, I hired them then in my other ventures. And basically the systems, the methods, the approaches that were used under MIB were then redirected and in order to build a system that is applicable to drug development in the context of inflammatory and autoimmune disease. So if you think about what Titan X is today, it's a combination of bioinformatics, advanced computational modeling, and advanced AI algorithms that work together in order to address specific questions, from understanding the inner workings of a CD40 cell to understanding how certain drugs will operate in the gastrointestinal tract, to making predictions on clinical trials based on certain databases. So it's work in progress. I think that we will never complete the development of Titan X. It will have version 1, version 2, version 3, version 23. And it has been absolutely instrumental in how we have done target discovery for novel immunoregulatory pathways, how we've characterized some of these pathways, how we've designed the mechanistic studies, and how we've run the translational and clinical trials for omilancor, for NIM1324, for NX13. And we continue to utilize Titan X as we continue to build Titan X with novel modules. I think the latest module that we've established is what's called the M2 pipeline, modeling metabolism pipeline, which enables us to characterize the changes in metabolism going on in each of the in silico immune cells within the Titan X platform.
So how do you, because that's usually what investors ask, or skeptics, I guess, about the AI aspect, is like, how do you validate Titan X, prospectively, or how do you run blinded checks to make sure it's actually predictive?
Yeah, that's very important. And that was a very deliberate decision that I made in 2010 when I was the director of that NIH-funded program. And I've continued that approach since then in my different biotech ventures. We never believe a prediction derived from computational methods unless we've experimentally validated. So there will be some predictions that will require going back to the lab and running in vitro experiments. In some cases, it requires doing mechanistic animal studies related to immunology. In some cases, you need to do translational work with human cells. So I think that even when we were publishing papers about this, now from 2010 until 2015, we did not publish a single paper related to computational methods unless we had the prediction and the experimental validation of that prediction. From that perspective, we've envisioned what we generate using AI, advanced computational modeling, and bioinformatics as almost an engineering cycle, where you build a model, you calibrate the model, you generate novel predictions. Maybe the most transformative of those predictions are worthwhile testing experimentally. Then you go back to the lab, you test. And then if it turns out that that prediction is not correct, then what you need to ask is, what's wrong with the model? What the specific parameters need to be modulated, modified, so that when the model runs again, the prediction is correct. And if it turns out to be correct, then it becomes part of the model as a further validation. So it's an iterative cycle that keeps going on, and every cycle of simulation and experimental validation, you are strengthening your database, you are strengthening your modeling capabilities, and you are strengthening the predictive power of your tool.
Thanks for explaining this to our audience. It's always this, it's kind of in biology, you always have tools that help you in some ways. And I think AI is really enabling to go much faster, right? It helps you identify something, but then obviously you can't just take it for granted. You have to validate. And so thanks for explaining how you kind of started that. And it's very fascinating.
My pleasure. And I think at the practical level, if you put numbers to the use of AI, you can go in a short period of time, you can go from a pipeline of 0 drug candidates to a pipeline of 20-some drug candidates with different levels of validation, preclinical and clinical, in a period of time that could not have been possible, let's say, in the early 2000s.
Exactly. So before we wrap up, I just have 2 quick questions. Feel free to answer those as much or as little as you want. But what do you think is a mistake that smart biotech founders are still making?
Oh, look, I think that there's not such a thing as the infallibility of biotech founders. I think that we, including myself, tend to be very faulty, as opposed to the Pope that defines himself as infallible based on doctrine. We shouldn't do that. Look, I think that one of the key mistakes is identifying the wrong partners. Right? Sometimes you are so committed to advance that technology that you really want the resources to be able to implement. Clinical trials are expensive. It's not that you can support all of that with just grants and contracts and non-dilutive funding. So you have a need for capital, which is significant. And sometimes the drive to get to the next level, to go from IND enabling to phase 1, from phase 1 to phase 2, phase 2 to phase 3, sometimes you just want to close things, and then maybe you fail to run enough diligence about who your partners are going to be, because let's be honest, this is kind of like a marriage. You have the technology, you have the company, but once you choose certain partners, they become part of the board, they influence the direction of the company. So what I would say is sometimes it's best to take extra time, do enough diligence about who your potential partners are going to be, and make the right decisions. Because if you make a mistake at this level, it has the potential to come back to haunt you later on. And I'm sure there's many other mistakes that founders make, but this is a quite consequential one.
No, thanks. I talk to a lot of founders, and usually when I ask them this question, I would say 80% will say this as a first thing is if you surround yourself with, it's kind of exactly like you said, like a marriage, right? You have to surround yourself with people that will help you and hold you accountable also, and then enable this partnership to go further and achieve great things. And then lastly, I just, in 1, 2, 3 sentences, where do you see your company in 5 years from today?
Yeah. So look, right now we are probably the only private company that has a phase 3 asset in ulcerative colitis and Crohn's disease. We are hoping to have data readout within the next couple of years for our phase 3 clinical trial. We could be setting our company up for commercialization, so that's a potential outcome. Or we could be acquired by a big pharma. And in the end, we'll make the decision that is going to be best for our shareholders, for our employees, for the patients, and I'll contribute to making that decision. I think that those are the likely 2 scenarios. In between, whether we'll remain private or will change, I think that's to be defined. But 5 years from now, I think that those are the 2 scenarios.
All right. Thanks, Josep. This was great. Thanks for taking the time, to talk to me today. And for our listeners, I'll drop links to NImmune and Josep's, LinkedIn and stuff in the show notes. If you enjoyed this conversation, please share it with a founder or a friend or a colleague. And thank you so much, and we'll see you next time.
Thank you very much. Thanks for having me.
Thanks.